Welcome everybody to another mini Beyond Terrain lecture. Today we're going to be discussing viruses, everyone's favorite topic nowadays. And there seems to be a resurgence of the discussion on exosomes, which we'll cover slightly in this episode. But we're mostly going to cover why viruses do not exist in this episode. I'll be touching on the resurgence of the exosome theory moving forward. I'm going to do a couple review videos as well as some original videos and some content on that. So if you're interested, make sure you let me know and I can get those videos out. When it comes to the existence of viruses, there are essentially four types of experiments that are run to prove the existence of them. The first one is cytopathic effects studies. The second is genetics. The third is immunological studies, antibodies. And lastly, you have electron microscopy. We're going to cover these four briefly here now. And if you're interested in this topic, I wrote a fairly comprehensive paper on this topic, going over the methodologies used more in depth, where we covered the specific experiments and tools used to prove the existence of a virus. Here we're going to cover it briefly. We're not going to be providing any references, of course. It's video format, but you can go check that out and get a more comprehensive understanding of the argument and the tools used in virology. So the first one, we have cytopathic effects studies. Largely, the important thing to stress here is that virology touts that they have an isolated virus that they introduce into a cell culture, which in turn kills some cells and produces a visual effect on the cell plates. The main problem here is that viruses cannot be isolated in a purity, right? And even a 99% pure sample is not achieving the standard that the logic behind empiricism uses to employ the scientific method. So within the cytopathic effects studies, essentially what's being done is they're taking a concoction which may or may not contain a viral particle, right? But it also has a plethora of other chemicals which they introduce into a cell culture and they observe the effects. Now problematically here, they use a control called a mock infection. Now the mock infection should have the exact same chemicals in it minus this so-called virus. Dr. Stefan Lanka has shown in his experiments, when it's properly controlled, the control cells end up dying as well, because it's the medium that the so-called viral particle is in that's causing the damage to the cells. Often they use viro cells, monkey kidney cells, and they introduce streptomycin, an antibiotic, which is a known toxin to viro cells. So of course you see cell death and you see cellular breakdown, maybe through electron microscopy, but essentially that's the thing here, right? So there's no actual virus. Now, this is where the discussion of exosomes come in. What's being observed through electron microscopy? You see these little particles, viral-like particles they're called. Some call them exosomes, extracellular vesicles. They're just small little vesicles. They're smaller versions of cells in a way. They may or may not contain genetic material. They may or may not contain proteins on the outsides. So let's move into electron microscopy more specifically here. Of course, electron microscopy has many problems within itself, and so, it's certainly an uncontrolled endeavor. There are many steps that are conducted, such as staining with heavy metals, dehydrating, rehydrating with some sort of artificial hydrant. You have to dissect it, you have to cut really small, thin samples of it, you have to mount it on a plate, it needs to be dehydrated, frozen, etc., right? So there are many steps that go uncontrolled. So electron microscopy in itself is largely uncontrolled. Now, if you take cells and you take electron microscopy pictures of cells with nothing introduced, healthy cells, you won't see virus-like particles. But if you take diseased cells or stressed out cells, you do see virus-like particles. So there may be an important distinction there in the discussion of exosomes, whether or not there are meaningful cellular breakdowns which play a process in the body. That we're not going to discuss right now, because we have to take it one step back here. Can we actually prove that these cellular breakdowns are not laboratory artifacts? And so I'm not opposed to there being this sort of vesicle that breaks down in the body, especially in times of stress. It seems rather meaningful, and if you take an alchemical lens, it's a microcosm, say, of a larger process in the body. So it might have a lot of validity to it. That being said, to make any claims about these particles, I do believe is impossible, especially about their processes, since you cannot isolate them, and this is admitted by virology. The process of isolation is extracting a virus from a healthy individual and introducing it into a cell culture. right? There's never a time where it's pure. There's never a purity. So to understand anything about the nature of this is really, really difficult and nearly impossible until you can achieve that purity. The purity, the isolation, is a necessary component of the scientific method. It's the isolation of variables. So we're all over the place here. So back to electron microscopy. So the process in itself is an uncontrolled endeavor. The other problem is, again, back to that mock infection. There are no true mock infections being done, especially with electron microscopy. The other problem is that it's impossible to distinguish any sort of viral particle from other particles in the body, if there are particles in the body. So it's impossible to make a positive claim about some sort of specificity about the particles on the nanoscale, right? There's no way of truly separating viruses from viral-like particle, exosomes, vesicles, etc., right? So to be clear here, the entity virus, we deny. I'm going to sit here and say that it's possible that there is a particle that is created by our cells during cellular stress and disease processes and breakdown. Because, of course, through electron microscopy, you could see these structures in cancer cells. But it's really important to understand that the term virus, meaning this self-replicating particle that can replicate in your cells and hijack your cellular mechanisms to produce disease, is what we are truly against here. And so I really want that to be clear. So I'll try and keep this as brief as I can. When it comes to the genetic analysis, there's never a purity of genetic material, and so the methods in which they use these shotgun sequences or metagenomics, where they're processing large amounts of genetic material from an unpurified sample and creating it in a computer through probability, there's no control here, whatever. So it's certainly not a scientific process. There's no controls, there's no isolation. You cannot control the processes used in New Age genomics, and you cannot use the gold standard Sanger sequencing, which still has its problems, but you cannot use Sanger because Sanger requires isolated genetic material, which again you cannot do with a virus, because the only way to isolate a virus is to have it in a cell culture, and we know that cells carry their own genetic material, and there might be other contaminants of genetic material as well. Even if you take a swab out of somebody's nose, even in your mucus and your saliva and any sort of bodily secretions, there are genetic materials. So that's a really important distinguish. Genomics, especially when it comes to virology, is largely an uncontrolled endeavor. And so understanding that is fundamentally one of the most important things to understand about the virology debate here when it comes to no virus. Lastly, we have the immunological assays. This comes things like ELISA, SDS page, etc., etc. There are many different ways to run antibody tests, but the general idea is that an antibody can detect an antigen, and these are proteins. So some sort of antibody can detect the protein on a virus and detect its presence, right? Because you can use fluorescence, you can use different methodologies to prove that that antigen or antibody is present in the sample, or that it's activated and binded to a certain protein in question. The problem with antibodies is that all antibodies display some sort of non-specific behavior. So again, it goes largely uncontrolled in its experiments, but philosophically speaking, when it comes to the theory of antibodies, there are no specific antibodies. There are 5 big ones that you're taught, and then we say that they all have some subset of specificity, but any sort of experiments that try to prove specificity of antibodies fail, right? They often can bind to many different bands in these experiments, so they're non-specific in nature. Again, they go largely uncontrolled in experiments where they don't run it against samples with other genetic materials or other so-called viruses or particles. So you're missing a really fundamental part of the scientific method here, where it's uncontrolled. And the underlying philosophy of antibodies, of course, falls short, because there's truly no specificity, and even a positive read on one of these doesn't necessarily indicate that the specific antigen was present in the sample, because you don't know whether or not you're acquiring the right binding. It can bind to not necessarily anything, but a large subset of things. And there are lots of problems with antibody research that's admitted in this. There are false positives and false negatives all the time in antibody research. And even acquiring any sort of purity of antibodies is, again, a very, very difficult and may be impossible based on our understandings and methods at this point in time. So my final thoughts on this essentially are that viruses have never been proven to exist as self-replicating particles which can hijack your cells and cause disease. They've certainly not been shown to prove any sort of causative agents in disease, because contagion studies constantly fail when they are controlled whatsoever. The real problem with virology is that it's completely undertaken a circular reasoning. The cytopathic effects rely on electron microscopy, immunology, as well as genetics. But the problem is, to prove that we've understood the genetics, it requires us to use cytopathic effects studies, electron microscopy, as well as immunology. And to prove immunology correct, it takes genetic studies, electron microscopy, cytopathic effects. And to prove the electron microscopy, to show that there is a virus in the sample and not just a virus-like particle or some arbitrary little particulate in the cell, it requires genetic analysis, immunology, as well as cytopathic effects studies. And so they all rely on each other. This is a very intricate form of circular reasoning, but when there's no beginning and no end to the string of logic, that's when it breaks down. So that's a really important thing to understand about virology, is that it is circular in nature. Genetics is largely circular as well, especially when it comes to virology, this new age metagenomics and anything in silico, using the computers. You know, it's probability based. It's created by human beings with our own dogmas, with our own understandings of reality. That's what's interjected into these softwares. So there's a great bias going on there. And Thomas Kuhn would certainly have lots to say about this as well. He already argued that the worldview of the individual can shape the observations being made in empiricism. And so that's really important to understand as well. So of course we're going to discuss exosomes moving forward. We're going to discuss the probable presence of a cellular breakdown in the body. It seems very probable, but what do we know about cellular biology? We'll delve into Harold Hillman's works and things of this, but let me know exactly what you want to know about, any questions you got down below. If you enjoyed this, like, share, comment. Sharing is the best way. There's links if you want to go further there, but getting the word out and sharing to a friend. I really want to get this information out there to a lot of people. And so if you enjoyed it, make sure you do that. We're giving a book away, so remember that. And thanks for listening. Take care.